the first paper is looking at prophylactic possibilities. Um, this is the idea that you can either immediately, before infection or immediately afterwards, you can stem the progression of the disease by offering some therapies. The second paper is a running meta-analysis. So every time there's a new study, it's added into this. And there's a really cool interactive figure that we wanted to go through where we can sort of compare the efficacy of different treatment regimes. It runs into the same problem that I was talking about earlier, where the standard of care is a moving target, and that seems to be the benchmark for whether something is working or not. A lot of the combinations that they're trying out include items that are on the standard care, so that complicates things further too.
All right. So one of the things that really astounds me, given how much time and effort and energy has gone into researching COVID, treating COVID, figuring out what is going on with COVID, the still somewhat shaky data on what really, 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 really works. From that BMJ study, which basically ran through all of the different treatment options and gave you odds ratios for all of these different things, the bottom line seemed to be that the only thing that really worked was suppressing the immune system. Would you agree with that?
Yeah, and I guess I'm gonna keep beating on this dead horse, but if your comparison in a study is that you have two groups of patients and you give one patient a medicine and you give the other patient water, but you tell them it's a medicine, that's a pretty good study and you can probably trust what you get out of it. If you give one person medicine and you give the other person a new medicine every single time that you have a treatment, you don't really have anything to compare it against. And that's, I think, why we're not able to pull anything meaningful out of this data. That standard therapy slash control is a moving target. It's changing constantly. There's a running list at the CDC of the standard therapy. Oftentimes, it includes drugs that are in those experimental categories too, which I can't even imagine how you deconvolve that.
You would have to pick the studies that you look at to make sure that you're only, because I mean, each study will list what its standard of care is. And so you'd have to go into each and every single one and make sure that,
Like this is today's as of March. I think this is listed as March 2nd is the last time they updated it. This is the list right here. Preferred therapies listed in order of preference. So already the control group has any one of these drugs in it. But if they don't have it, then you get these other drugs. Now, they're sort of similar, right? You have a protease inhibitor combo up front. That's what this nirmatrelvir and ritonavir is. You have a monoclonal antibody there, which is Sotrovimab, and remdesivir, which we just talked about.
which is given its complicated nature of increasing the likelihood of hospitalization, it's amazing that it is listed as a preferred therapy because that suggests that the only better thing that they have is this new Pfizer drug, which is called Paxlovid.
Which is also a protease inhibitor, I believe.
Mm-hmm.
Okay. So, but they're saying that standard of care is this stuff, but it could also be this stuff down here at the bottom, which is other versions that are sort of similarly acting. As some other sort of protease inhibitor,
Right. So I looked up.
a
There
different kind
doesn't.
of monoclonal antibody.
Yeah, there doesn't seem to be much of a difference between these two different monoclonal antibodies. They're just produced with slightly different animals, I think.
But if you go to, can we just click on this link? Is that possible? Does it take us there? No. If you go to this link down here, you go to the CDC website, you'll see that this list of, uh, you'll
Mm-mm.
see, no, it's okay. You'll see that the list of standard therapy has been shifting throughout this pandemic. How that factors into your, your studies comparison, I have no idea.
I mean, you have to, this is the great difficulty of meta-analysis, is that when you go to do a comparison, you have to figure out, if you want to look to see what remdesivir is doing, the way that the BMJ article is organized is that they're looking at either standard of care or placebo, right? And so that means that if they're looking at remdesivir alone, they're not necessarily comparing it to standard of care, which is a different drug. They might be comparing it to placebo, because it's given all by itself and they're just looking at remdesivir. But then you start to have other complications, because when you're doing clinical trials, you have a question of ethics. And the question of ethics is this. You have in front of you a patient, multiple patients, many patients, 10,000 patients. And you have to break them into groups. And one of the groups is going to get a treatment, and the other group is going to get not the treatment. And you have to sit down and you have to ask yourself, okay, well, what can I give to this not treatment group, which would still allow me to evaluate the efficacy of my treatment, but not end up killing these people or maiming them or significantly damaging them, even when there is a better treatment than nothing that's available, right? So let's say that you have drug A, which is the standard of care, and you can't withhold drug A from a population that's highly at risk of some disease. You have to give even your placebo arm drug A, because it's unethical to withhold treatment that you know would work. And this complicates things further, because if you start to go down in age, you get to a point where it might actually be ethical to give a group of people that you know don't get severe COVID no treatment. And then you would actually be able to compare the effects of your drug to a placebo, because you make the case that these people aren't going to die of COVID anyways. I can actually make this comparison and it won't be evil. And your review board gives you the green light and everybody's like, okay, fine, they're going to be okay. We're going to look to see how remdesivir works. But you can't actually extrapolate that to how remdesivir will work in 80-year-olds. You have to actually look in 80-year-olds, because there's so much that happens to the immune system and to the body from the age of 20 to the age of 80 that comparison becomes almost impossible. And so when people do meta-analysis, you trust that they have done these comparisons carefully, but always take it with a grain of salt, because there's no bottom to this. There's no hard basement bedrock that you can get to.
I mean, I think it's also worth asking who's doing these studies. Like, what is a clinical trial? How is that happening? And the answer is that 6 out of 7 times it's a private company that's paying for those clinical trials. So they're pushing them through the whole adjudication process too, from start to finish. They're designing the trial and they're paying for it to happen all the way through. And it's almost impossible to imagine separating out their particular interest in seeing their drug be looked at least. I'm not even saying that they're playing for their drug to come out favorably, but like the selection of drugs which is getting looked at are the ones that are being paid for to be studied in the first place.
And so there's a huge push behind the fact that they're studying them, and it's very difficult to know when you're looking at clinical trials, is there good data or is there not? Like, for example, one of the things is statins. Statins are drugs that are given for high blood pressure, and they are prescribed basically as a prophylactic to everybody over the age of 50, probably. Once you get to the point where you have high blood pressure because of the fact that you've aged into this category, your doctor will write you a script for statins. The problem with getting a prescription for statins is that it doesn't appear to decrease mortality. So you're getting a drug that has been approved by the FDA for treating a condition which is high blood pressure, but it doesn't seem to actually help with your health. It brings down high blood pressure, but it doesn't help you sufficiently to actually prevent mortality in this case. And so it becomes very, very difficult to assess all of this.
They keep flip-flopping on that, by the way, and they're like pulling out individual risk groups that can maybe benefit from it. But for many, many, many years, it was the case that statins didn't really seem to do anything. I mean, they do bring down your blood pressure. But
so has this been overturned since the last time that I looked at it? Do we now?
I've seen some headlines where they're like this one, among individuals with a history of clinical coronary heart disease, all-cause mortality was reduced. I don't know how much or whatever.
Okay.
But
So
probably not much.
basically, what happens is that you get a drug that is brought to people and is told that, look, this is going to help everybody. It goes out into the field. After some years, they realize that it doesn't necessarily help everybody, and then you start to chip away at the sort of the full picture in order to be able to carve out something that actually is really robust. And these processes can take decades. Statins have been around since the 70s.
I don't know.
Statins have been around for a really long time.
I mean, this is such a fascinating systemic quagmire, because on one hand, the private industry has accelerated the development of all these amazing technologies, but at the same time, there's no overarching organization of these trials, right? It's all completely, each trial is independent of the other one. There's no central processing place. There's nobody going around to make sure that everybody's doing it the same. There's not even a standard control in all these trials, right? So it's like, it's kind of the wild, wild west out there. And this is a real problem that's going to have to be, you guys are going to have to figure this out in the future.
I mean, everybody has to figure this out.
Yeah.
I think that we're putting too much pressure on the next generation.
I'm like, you kids can do it.
It's like
We're
one
too
foot in the
old.
grave. It's all your responsibility.
But yeah, it's a real interesting moment here. And of course, we've been at molecular medicine for like less than 100 years, so don't panic. It's just the early days. We're not quite space age yet, kids.
So when clinical trials are done, they usually happen in 4 phases. So the early discovery and ongoing research is the realm in which I spent my entire career in laboratories. I never worked in the clinical side of things. I was always focused on super basic research. We were looking in mice, we were looking in cells, we never dealt with patient data. And this is the sort of work that is done at the bench, which, if it is promising under bench conditions, then if
this is like ivermectin kills COVID in cells in a petri dish kind of stuff.
Exactly. At that point,
10,000 times the concentration a human can tolerate.
And at that point, once that rises to the level of, oh, that's interesting, maybe there's something there, you go to phase 1. Phase 1, phase 2, phase 3, phase 4. These are all expanding the size of your treatment group. You're looking to see at each stage to make sure that this drug that is experimental, that you're going to give to people, is not going to cause them to have massive anaphylactic reactions, whole body autoimmune reactions. It's not going to fry their livers. It's not going to do anything horrific to them, because oftentimes it does. There's, you can actually be a guinea pig for these phase 1 clinical trials because they pay people money.
I was going to say in cancer, it's really, really, really bad. Like something like 90+% of the drugs that are developed in this microscope phase 0 don't work once they get to phase 2, 1, 2. I guess they don't even look for effects until phase 2. But 90+% of the molecules that work against mice in little cages do nothing against humans. So that's a huge weeding out phase right there.
Well, there's also the phase 1 the phase 1 step is well before data is collected. So by the time that the clinical trial is completed, 90% of drugs that showed promise in mice in the laboratory will have done nothing in human bodies. But even before that point, you can have pretty significant problems with safety and dosage levels. I don't know if you remember when the vaccines were first starting to come out, they were recruiting volunteers for this phase 1 setting, which is that people just showed up and they enrolled in this clinical trial and they got a small group and they just tested the vaccines in them to make sure that there weren't going to be any severe reactions before they moved on to the next phase, which is that you get basically a larger group of people. And then you get, you basically do a small trial where you have less than 100 people and you just basically give people the treatment. It's not super highly controlled. You might not even have a placebo arm. You are not necessarily double-blind clinical trial. That's not until phase 3. You first have to show that there is some basic promise that is going to lead you to get this next phase, which is hundreds of participants. It's enormous amounts of money. The pipeline of how the number of drugs that are candidates for each of these stages narrows dramatically with each phase, because you can get to phase 2 and you can give people the substance and there's just absolutely no effect. And if there's no dramatic effect, then there doesn't seem to be any point to getting to phase 3. This is probably the point where they basically bring people in and they just give them the drug and they see if they do better before they add any of the controlled settings in, right? Where it's not double-blinded. It's just very rough.
Very few are double-blinded anyways.
Very few are double-blinded anyways. Phase 3, regulatory approval. This is the stage that demonstrates that everything is working. And then phase 4 is basically after you get approved, then you get to market your treatment and you give it to thousands and thousands of people. And this is basically where we are with the vaccines. They've made it through all 3 of these phases and now they've gotten final full approval from the FDA. And so now they are past approval. So we talked a little bit about these standards of care. These standards of care would have been developed for other epidemics of SARS, MERS, other coronaviruses, influenza. Some of the things that we looked at were used for treatments none of the standard of cares. The standard of cares were developed in adjacent realms and then basically decided upon as being the best option for this. Would you agree?
I think it's just rolling. They're developing it as they go. It's also worth mentioning that standard of care is particular to a given pathology, and that during the pandemic, those other standards of care have been shifting too in response to overwhelmed hospitals, overwhelmed staff. And so a lot of other things like, say, stage 3 colon cancer is going to have a different standard of care. It's called a crisis standard of care during the pandemic than it would have otherwise. And so there's been a lot of writing in the literature about this with respect to what are the ethics of shifting the standard of care versus, what are the fallout casualties as a result of the shunting of all these attentions to COVID away from, because that's not even really been factored into people's mortality counts or morbidity counts and stuff like that.
Definitely. All right, what's this?
What is this? Okay, so this is kind of interesting too. I just want to really make sure that we understand this idea. This is, I think, the most important idea in all of this is that all these drugs are being evaluated against the standard of care that's shifting. This is the standard of care for patients who are not hospitalized. The next slide has the standard of care for patients that are hospitalized. And they're very different, and they're on the move also. So you can see a number of these drugs that were on the previous slide.
Mm-hmm. So Paxlovid is, again, basically the standard of care at this point.
They have their own schedules, right? Like they're still recommending remdesivir, but as soon as you're discharged, stop taking it, right? So maybe they're hoping to pull back on some of those negative effects that way.
Yeah, I mean, you should not send people home with a dexamethasone prescription and just be like, take 2 and call me and see you in a week or something. These are pretty powerful drugs that can cause significant
peripheral problems too, right?
Yeah.
If you get knee surgery or something, you might get some steroids and it'll help the swelling and stuff like that. But you might come down with a really nasty pneumonia or something because they're very broad-based immune inhibitors. They literally just tell your immune system to go to sleep, and that's probably not what you want to have happen. You could probably end up with much worse things down the line from that if you took it over and over again. You could end up with cancers and things like that.
Mm-hmm.
So this is for hospitalized patients, and again, the standard of care.
Oh, hold on. I'm sorry. Can we go back just one?
Yeah.
I was looking at these, and so these all have these ALLA, BLLA, and I was trying to figure out where these are actually 2s. So it's A2A, B2A, and if you look down here, there's a legend. So the rating of recommendation is A, B, C, which is strong, moderate, or optional. And then the rating of the evidence has to do with either one or more randomized trials without major limitations, a larger test of randomized trials, non-randomized trials or observational cohort studies, and then the third is expert opinion. And it's very interesting that for most of these, these are kind of given with some caveats. Not a single one on this page of recommendations is a strong recommendation that also happens to have a very strong randomized controlled trial without major limitations. They're all other randomized trials or some group analyses of randomized trials. So it's basically they're mining the data from other trials in order to be able to find the justification for prescribing these drugs. And then there's also some of these that are just expert opinion, right? So symptomatic management, that's a strong recommendation, and that's a strong recommendation on the basis of expert opinion. Corticosteroids being recommended against is a strong recommendation on the basis of expert opinion. This isn't stuff that has clinical trial data behind it. Okay. Dexamethasone, again, is a very scary thing because it really, really does control your immune system very effectively.
It's a steroid.
Mm-hmm.
So it's not surprising that it works at controlling inflammation. The question is, what other bad stuff happens as a result of that that's not included in our metrics here? What sort of malignancies will show up later down the road? What sort of heart disease will show up as a result of that? What sort of autoimmune disorders? These are difficult questions if you're not specifically asking them, and the likelihood is that no one's specifically asking them because it's gonna be a tricky thing to pay for.
And here we're starting to see that in more severe conditions, we're breaking out the recommendations for therapy between antiviral and immunomodulation versus anticoagulation. We've talked in other lectures about the role that COVID has in activating these clotting cascades and that it is a huge clotting problem throughout the entire body. So it makes sense that by the time that you get hospitalized, that you're giving a prophylactic dose of heparin. And this is actually supported by direct clinical trial evidence, where if you are at the point where you are unhealthy enough from COVID to be hospitalized, we have to be on the lookout for making sure that you're getting the heparin that you need, because if you don't, you can have significant clotting effects.
Yeah, they didn't really mention the anticoagulant side of things in these papers, which is interesting. Actually, it might have been in the interactive graphic. You want to just jump over and check that out real quick?
Let's finish walking through this.
Because we haven't really even talked about the classes of therapeutics yet.
Sure.
Maybe that's worth at least mentioning.
I agree. Basically, this is just a continuous increase in severity of COVID, right? So you hospitalized but does not require supplemental oxygen, goes all the way to hospitalized and requires extracorporeal membrane oxygenation or mechanical ventilation, both of which are very, very severe interventions. By the time that you get to the severe intervention, we're going corticosteroids plus monoclonal antibodies. And if there's not this specific brand of monoclonal antibodies, then use a different one. But that's basically the standard of care because this seems to be the only thing that's helping people.
So if in your meta-analysis you're comparing some new drug against the standard of care, you don't really know which one of these you're looking at is the problem.
Well, the people who did the meta-analysis
did. But we don't know that they're comparing it based on age groups and stuff like that.
The people who did the meta-analysis do, but it's hidden for us. Yeah. And it doesn't seem to be broken up by age, and that's a really confounding thing because if you tell somebody, if you look at something like heart disease, you can tell somebody that, okay, so if you're above the age of 65, you're at a high risk for heart disease. And that is true relative to somebody who's 20, but not every single person over the age of 65 has the exact same risk for heart disease. If you take care of your body, if you eat well, if you have a low stress lifestyle, whatever, your likelihood of, if you're rich, right? Your likelihood of having heart disease is lower than if you don't have any of these things and you have pre-existing conditions. And so it's very, very unclear
The average doesn't always account for the individuals. That's why you have to be really careful when applying these statistics to
Mm-hmm.
yourself, right? You guys are technically, I could say to you, you have a very high risk of dying from dying in a car wreck based on your age. But if you don't drive or go ride with people in cars ever, your chance drops to zero pretty quickly. So it can be both true that you have a high risk and completely untrue at the same time. It's very interesting.
Unless you take what that movie, what was it, Final Destination said, which is that you're fated to go one way or another.
You will end up in a car somehow.
Yeah, exactly.
But that's what's really interesting is that statistics can only put you in the ballpark, and they might be completely off if you have some sort of confounding issues, like you don't drive cars.
All right, this can before we move back to the paper.
Let's come back to that later.
Let's come back to this later. Okay.
Oh, wait.
Are you sure?
This is fine. I already mentioned this. This is just the idea that the standards of care are shifting for other diseases. We talked about that. This is just a headline from the NIH, who's the CDC. So, it's a real nightmare for them.
Aha. So this figure was the reason that I really wanted everyone to look at it. Which actually at this point is that true? It's almost a year old at this point.
That explains a lot.
Mm-hmm. Interesting. I keep forgetting that it's not 2021 anymore. However, I think that this does still hold because we go to the CDC website and it has the same data. I think that they stopped updating it because it wasn't necessarily changing anymore. All right, so what do you want to lead us through this?
Oh, I just thought we'd just skip around and see what's up. So this is, I hope. What's wrong?
I didn't realize that you could also add the very, very low,
Hmm.
and then it turns into basically sacred geometry.
Yeah. This is, I hope, the way that scientific reporting goes in the future. I hope that we have better data than this, but this is really cool because we can actually ask questions ourselves. For instance, mortality. What sort of, no, that's interesting the way it was. Leave it there. Sorry. I thought it was interesting. What can we say about mortality as of April 2021 with regard to all of these therapies? What can we say about it that is highly supported by evidence? And the answer is absolutely nothing. And that's okay, because that allows us to make decisions with some level of uncertainty that we might have to change our mind down the line. And that's why these therapies keep changing. If you click on moderate, can you add moderate in? Okay. If we say that we want to look for moderate certainty that there's an effect going on, now all of a sudden there's this slight increase here with corticosteroids. And this is pretty cool. So what do we have here?
So you basically have a decrease of you have, if you're taking corticosteroids, you have 80% the likelihood that you will end up dying of COVID than if you were taking whatever standard of care is in this case, which is either standard of care or placebo. Standard of care can be any of the drugs that we listed or whatever was on the schedule in,
I tried really hard to find what was on the schedule in April of 2021, and I just couldn't get there.
Took us.
Sorry.
So then what we see here is that the majority of the interactions are very low or are low, and then even more of them are very, very low. And so you're starting to see that these are effects that are imprecise, that they're kind of washed out when you start to look at all of the data, that it's not carrying through. They won't even really let you look at it too specifically.
At least so mortality data is pretty bad. But what's cool is that if we go and look at something like ventilation, or when we're in the hospital, if any of you go into healthcare, there's a lot of decisions that have to be made at the point of patient care. And this is how those decisions are made. So can we look at
I'm trying to see if there's anything that has a high correlation.
What do they mean by high, by the way?
It's just that they think...
They probably tell us in the how to read this diagram or something.
They really do... It's an evaluation as to how convincing that they think that the data is.
Right, but I bet it's based on some confidence interval or something.
So it seems to be that a year ago, there's absolutely nothing that had any kind of high evidence quality. Sorry, I'm pulling on your headphones.
Oh, here we go. We'd have to read the body of the paper to see how many times. They usually tell you exactly what the odds ratios are when you click on the dot. Yeah.
Yeah.
Or on the, is it on the
Well, this is
Murphy?
ventilator-free days, so
Oh.
you're looking for like mechanical ventilation.
Yeah, so you're trying to make a decision. How do we only have so many ventilation machines, ventilators? What therapy should we implement to minimize the necessity of that? And it seems like we have all of one option here.
And this is a huge part of why there was an enormous push for vaccines, because it basically was starkly obvious that when you put people down in front of the data, that there wasn't a lot of super high, very, very reproducible data that gave people the ability to say with certainty that some treatment was going to work. Vaccines, though, were presented as being functionally effective, right? It was the promise that once a vaccine was developed, it would allow the pandemic to end. And you heard this from much before April of 2021, where there was the last great hope that the vaccines would allow things to return to normal. And I think that much of it was driven because the data was so spotty. And as we saw at the website that we pulled up at the beginning, which is this ivermectin and everything else website, that you have a lot more data now about the various treatments and how they actually work and if they work. But there's not necessarily a guarantee that this is the same for all variants of COVID either. And so this is actually quite shocking, where this is the Sotrovimab, which is one of the standard of care that's being promoted by the CDC, but there's not really a clear and cohesive indication that it works in late treatment. It only works in early treatment.
And even then, it's a pretty huge spread.
It's a pretty huge spread. So what do we have to say about the BMJ article?
Oh, I just think I think this is an interesting way of presenting and looking at data. So in that, before we leave mechanical ventilation, we can see that it's probably a good idea to put the patients on IL-6
inhibitor, IL-6 inhibitor.
IL-6 inhibitor. And it looks like corticosteroids do something too. And so by basically what they've done with this is they're taking all of these different studies that all of these different actors have put together and funded, and they've tried to make some sense out of it. And
they've come up with a shockingly low number of connections.
That's still something, right? This is still something.
But so what?
Even,
what I think that this is showing is that when you are looking at something that has to do with immune system dysfunction, the things that work across the board for everybody without any kind of nuance are the absolute brute force sledgehammers, right? So you give corticosteroids, it is a global drug that damps everything down. You give IL-6 inhibitor, global treatment that tamps everything down. If you start looking at what's showing up here, let's look at low rather than very low. What you start seeing when you start looking here is that you have other treatments that are not these global immune system modulators that are, like, for example, vitamin D. It might be a global immune system modulator in some people that have a high level of vitamin D receptor on the surface of their immune cells, but it's not the same as corticosteroid, which is basically taking a hammer to the system. You know what I mean?
Yeah, it's not clear that even all people are deficient in vitamin D anyways. For instance, if you live on the beach, you're probably doing just fine.
Exactly. And so it'd be impossible to compare. And if you go into the very low, then you're basically just attempting to tease out teeny tiny effects that might help some people and don't help other people.
Might also just reflect the differences in controls in each of those studies as well.
Right, exactly.
So it's static. Very pretty static, I must say. I mean, when you had the sacred geometry picture.
Yeah, seriously.
So
Quite.
I think, we just wanted to bring that paper in to hopefully look at the best way of analyzing a bunch of different studies all at once. The other paper is going to look at prophylactic treatments, and it's also going to take us through a lot of the main treatments too, and we'll get into the mechanisms because I want everybody to walk away from this lecture at least understanding the basic strategies for stopping the progression of a viral infection, COVID specifically. But almost all the therapies we're going to talk about today can be used for other viruses too. Um, Most of them apply to influenza. Several of them apply to HIV. So these are very broad antiviral treatment strategies. And although this paper is called potential prophylactic, they have a very loose definition of prophylaxis, which we usually think of as preventative. But like I said earlier, this is often about stopping a infection once it has started. And yeah, should we cruise through them?
Yeah, let's do it. Do you want to go to the slides, or you want to do the paper? Because we have a bunch of this in the
We
slides.
can bounce back and forth.
Okay.
Did you pull these into slides?
Many of them.
Maybe we can go through the slides then. Yeah, because I think they're in several broad classes. For instance, we have protease inhibitors and monoclonal antibodies.
Before we got into the body of the paper and we started talking about what it was that they were reporting in terms of treatments, I wanted to talk a little bit about the statistics that they're using. Hazard ratio and odds ratio are things that show up all of the time. And I think that it's important to have a working relationship to it. You'll see risk ratio as well, but if we didn't run across it in these two papers. So I didn't include it. But basically what you have is that your hazard ratio is it's a ratio. It's a ratio between the chance of an event happening in the treatment arm versus the chance of an event happening in the control arm.
So it's usually reported as a decimal place, though.
It is. So it's not presented as this fraction. It's given as 0.9 or 1.2 or whatever else.
Which is just like 102% or 90%.
120, right? 1.2.
90 to 1 or 100, sorry.
Well, so
it is a ratio.
If you have a higher so if you're looking for adverse events, if you're looking at, okay, I'm going to give a drug, I'm going to give a treatment, and I want to see what effect it has on mortality, the way that you'll know if it has an effect on mortality is that you're going to take the likelihood of the event happening in your treatment arm versus the likelihood of the event happening in your control arm, and then you will divide them, and then you will get a number. And that number will tell you if it happens more frequently in your treatment arm, that means that your treatment arm is killing people. If it happens less frequently in your treatment arm, it means that you are helping people. If you're exactly at 1, then that means that you're basically balanced and it's not different from your control, whatever it is that you're using.
Is this from the first paper?
This is from the BMJ paper. So the BMJ paper uses...
So if we didn't have that beautiful interactive graphic, we would be stuck sorting through these numbers instead.
And this is what I mean about the fact that presenting this in just words
Which is how it always used to be done. This is a brand new wave of visualization.
Right. So if you look at statistical papers, it was almost always presented where you just have to look at a table and you have to look at the numbers and you have to know what the numbers represent. And because you know what the numbers represent, then you're able to make some kind of conclusion. This isn't even presented in a table. It's literally the BMJ paper is just paragraph after paragraph of information about what these drugs do and how they work. And they're using a different marker, which is odds ratio. And so odds ratio is basically the likelihood of something happening in your experimental group versus the likelihood of something happening in your control group. And it's an average of those. And so it's very, very similar to your hazard ratio. But basically what you have here is that you have, and this is kind of sampled, it's not actually data, but you have an experimental group and you're looking to see in your final count, how many are sick, how many are healthy. In your control, how many are sick, how many are healthy. And then you make a ratio out of each one of these, and then you basically get your odds ratio. And it is the same it's similar to the hazard ratio in that if you have an odds ratio of 1, it means that you have no change. If you have an odds ratio that is less than 1, it means that you have a slight decrease. If you have an odds ratio greater than 1, it means that you're actually doing harm. If you look at these ratios here that they're talking about, the sort of the corticosteroids and remdesivir, you see that the spread is pretty big, right? So your corticosteroid spread goes from 0.69 to 0.98. So it basically goes from a pretty significant reduction to almost no reduction. For remdesivir, you go from significant reduction to actually causing harm. And this is often, so personally, I think that the reason that they report it this way, as opposed to drawing it out, is because if you actually drew it out, it would be obvious as to how bad the data was. Do you get that feeling ever?
Yeah, I mean, when you're working on this stuff, you want to report it. You want to give people hope, but this was in 2021 too, so there hasn't been a bigger meta-analysis that I'm aware of yet.
Mm-hmm.
And so it'll be interesting to see how this changes, 5 years from now. Of course, we're actually giving this at hopefully the tail end of this pandemic.
Mm.
And so we don't have all the data yet. I personally also just think it's these meta-analyses are fighting against the fact that there's no unified front in the way these studies are conducted or organized. And anyways, let's talk about all the different types of therapies. So there's a few broad classes. First one is monoclonal antibodies. We talked a bit about antibodies. I hope. Goodness. We did some immunology at the beginning of this course.
This is all immunology.
Well, we teach a bunch of classes. So we do teach an immunology course also, so I'm trying to keep that straight from
anatomy and physiology. This one's the immunology.
We taught in immunology last year. So.
That is true.
Okay, so all right, antibodies, right? They're produced by your own B cells. They are specific to something that your body has encountered, and they are essentially responsible for the memory portion of your immunity. When you get vaccinated against something or when you encounter it naturally, you make antibodies. This is the idea that you synthesize antibodies that are specifically aimed at particular parts of the virus, and you scale them up, you produce a ton of them, and you inject them directly into the patient to hopefully tag them for removal.
The way that we produce tons of them is that you take a mouse that has been injected with your antigen. So you take a mouse, you give it, you inject it with the SARS-CoV-2 virion, and then you isolate its spleen, and then you fuse myeloma, so these are cancerous cells, with the spleen cells to produce something that's called hybridomas. And once you've produced these hybridomas, you then culture these hybrid cells. The cancer cells will continue to grow in massive quantities. That's why you use a cancer cell, because it will never stop growing no matter how dense it gets. And then these cells, because they're immune cells, they are producing antibodies. And so you basically culture them to a massive degree, and then you break all of them open and you spin down the antibodies, and that's what's used as the medicine. When you're actually using it as a medicine, it is the same, in principle, the same as it is for your immune system, as if it was producing these antibodies, which is the fact that they bind to the surface of the spike protein somewhere here on the ACE2 receptor binding domain, which is at the N-terminal of this trimer. And then when they're bound, the immune system can come through and it can basically grab onto this squiggly little bit that hangs off the side, which is constant, and your immune system knows that it can recognize it, grab it, and get rid of stuff. If you give this treatment too late, it seems like you hurt the patient, right? So if we were to go back to this page where we were looking at all of these treatments, and you look at all of the...
Makes sense.
Right? So if you look at Sotrovimab, if you give late treatment, it either does nothing or it harms a little bit. If you look at...
Can we talk about why that is?
Please.
I don't want to cut you off, but...
No. And so this one's actually crazy. This other antibody seems like it
Wow.
does much, much worse.
Wow, that's really bad. I wouldn't have expected that much, but an antibody can only it can only really tag, it can only basically ramp up your immune system, right? It's gonna, if you have, if the virus hasn't already unfolded its program and started to derail the attention of your immune system and of your cells and started to actually clog up the gears, if you catch it early and you flag it for removal, then the antibody therapies make a lot of sense. If you've already started to suffer the collateral damage of having this nasty viral infection, if you're getting a lot of the scarring and the thrombic uh, symptoms and damage to your epithelial cells in your lungs, it's unlikely that pulling the viral particles, the virions, out of the solution with antibodies is really going to do much. Other than, if anything, it's going to excite the immune system even more because what do antibodies do? What happens when an antibody has tagged something in your body? Well, it's essentially a red flag that calls in other immune cells to come and eat it, basically, or to dispose of it in some other way, punch holes in it, basically do a bunch of work. So if your immune system's already completely exhausted and the rest of your body's torn up, and now you got these red flags popping up all of a sudden, telling the immune system to get back to work and deal with these cells, and it's already kind of dealt with them to the best of its ability. Uh, you could see how it would just, the immune system's just kind of pooped.
And it doesn't seem to be the case for all antibodies, and which is, I would think, a side effect of the formulation itself. So if you look at this, is Casirivimab and Indevimab. These don't. This is a blend of two different antibodies, and they don't seem to have the same effect. And so what I would think is that the Betalovimab has something that is wrong with its formulation.
Did they look at early versus late though in the other one?
They did, yeah. Early treatment, late treatment. Both of them are very effective. And it's actually more effective in late treatment than it is in early treatment, slightly. With a huge spread. So and the other thing to note is that with this Betalovimab, it's a single study that is still in a preprint, and it is from basically a month ago. And so it might be that this is really bad data still. And so you have to take all of these things into account when you look at a treatment. It's almost impossible to be able to just walk in, glance at it, and be able to come up with some kind of final decision that is going to be accurate for the long term. Other drugs that they said were really effective were these protease inhibitors.
Oh, yes. Protease inhibitors. All right, so this is actually getting into the paper. For whatever reason, the monoclonals didn't make it into this. I guess they're not prophylactic enough. So a major class of antiviral treatments in general seems to attack the replication of the virus, and in this case, um, there's several of these. What was the other one? Latavir. I don't know. There's a bunch of these. Ritanavir is the preferred one at the moment. To understand how they work, we have to go back and remember how the virion is assembled once it gets inside of the cell. And one of the things kind of interesting about this virus is that it makes this huge protein complex and then chops it up into the little bits that are capable of processing its RNA. And what a lot of these drugs do is they interfere with the chopping up of that big protein into the little functional proteins. And so you see these little yellow triangles here. Can you see hopefully they can see my cursor. Shows you all the places where this particular
Protease.
Yeah, protease, is doing its job to chop up this big protein, which is called the polyprotein.
So basically the way that it works is that when the virus gets into the cell and it decapsulates, it releases the nucleic acid information into the cytoplasm, you get these 1, 2, 3 open reading frames. But we know that there's a ton of different proteins that the virion produces. It doesn't just make 3 proteins. How does it do that? Well, the way that it does that is actually almost sickeningly effective. So if you look down here, this is basically ORF1A is broken out into this chart down here. And you can see that it starts from this non-structural protein. It's non-structural protein 1 is here, 2, 3, and then 4. And you have red arrows that cover the cleavage sites between these first 3. And then you have yellow arrows that cover the cleavage sites between non-structural protein 4 and 16. So what's happening? The thing that's happening here is that this first non-structural protein, the very, very first protein that the virion transcribes off of its RNA, is in fact it's also a protease. So what it's doing is that it's feeding back onto itself, and it's taking this long transcribed piece of RNA and it's cleaving the proteins that are coming off of it. And it's only capable of cleaving the first 1, 2, 3. So it basically can make more of itself, and then it can make 2 more, and then it produces this next protein, and the next protein is this 3C1 protease, which is capable of cleaving itself and everything else downstream. So it basically makes something that's very, very small, very, very effective to start the replication process going, and then it makes the super protein that's capable of cleaving everything else. And that super protein is the one that Ritanovir basically interrupts completely. And so when Ritanovir gets into the cell, it blocks its ability to synthesize all of these non-structural proteins. And if you look at what these non-structural proteins do, they're exclusively replicon proteins. They're there to make proteins, they're there to make nucleic acids, they're there to package things. We're not even getting close to structural proteins yet. These are all just nucleic acid proteins, and they make themselves. It's this ORF1 is a perfectly encapsulated replicon machinery that can make the tools for it to process itself. And if you can interrupt that, then you can interrupt the process of replication, and the immune system can actually get a handle on things. And we'll see this, one more thing, we'll see this over and over again, which is that any place where you can see a vulnerability in the virus is a way that you can treat it. If you can look at a specific aspect of its life cycle, if you look at a specific thing that it's doing, you can come in and you can just tweak that. And if you break the chain, it breaks everything because it doesn't have the ability to feed back and fix itself.
It's interesting because the paper that we assigned, the prophylactic paper, talks about these drugs, and it actually says that they don't work.
That's the combination of Lopanavir and Ritonavir.
Right. And so they do think that there might be some use in them as a prophylactic for healthcare workers if you give them before the patients are actually infected, which is quite interesting.
And so this is what Dr. Zlay is talking about. He's saying that basically if you look at...
Again, I think this plays into the idea that once the viral machinery has already done its damage and you have this massive immune cascade going on, the cat's out of the bag. Whereas if you can prevent it from happening in the first place with this antiviral program, great. But the virus doesn't appear, as we've already looked at in all this physiology so far, it's not like the virus is lysing your cells and that's your main problem. Your main problem has to do with the interaction between, really with how your body meets that virus and the inflammation that results from it, the coagulation that results from it, is really what's causing the majority of the morbidity and mortality.
Right, so it's basically saying that the drug concentrations that are used to treat HIV are too low in order to be able to deal with a lot of virus that's already in the body and replicating. But like you said, if you administer it ahead of time, because this paper is all about prophylactics, right? It's all about basically saying that, look, we have to get smart about the idea that we're going to send people into dangerous situations. They're going to be frontline workers. They're going to be in conditions where they need more protection than somebody else does. So how can we help them? And preloading them with a drug that stops viral replication before it begins means that you probably could stop a single virion or a couple of virions, even if you couldn't stop a full-blown infection.
I mean, yeah. It's also totally possible that your immune system could do it on its own and it just needs a little help, something like that.
Mm-hmm. All right, so corticosteroids. We talked
about these a little bit.
Yeah, we talked about it a little bit. I just wanted to show the sort of what the molecule looked like, and also show that there's multiple different ways in which corticosteroids end up being really effective. Where no matter what stage of disease that you are currently in, you're going to have a pretty strong reaction to corticosteroids because they're capable of stopping cytokine storms, which is what we're talking about, this massive immune reaction. They are capable of decreasing edema and various tissue swelling from tissue damage. If you ever get a high altitude pulmonary edema, or what does HACE stand for? Cerebral edema. So if you get high altitude pulmonary or cerebral edemas, the first line of treatment is dexamethasone, because what it does is it brings down the amount of extracellular fluid that's being pumped out of your cells, basically.
Or if you just get poison ivy.
Is that true?
I mean, we get hydrocortisone, right?
It's different from dexamethasone, I think.
It's the same.
But it's the same category.
It's still a glucocorticoid.
And then finally, it can also help clear with the fibrotic plaques. And so when you give somebody dexamethasone at all stages of treatment, what you're doing is you're basically giving something that will help recovery no matter where they are. And we have a question.
Yeah, is there anything that the body makes that's similar to dexamethasone, or is it like this completely exogenous thing?
Cortisol, right? Well, cortisol, it is a compound that does that. Dr. DeLay is nodding. So basically, cortisone is a stress hormone, and we see this that when you produce cortisol from the adrenal cortex that sits above the kidneys, it has very, very similar effects on the body. There's a lot of research recently that's been done for looking at how your ability to voluntarily trigger the cortisol system activates or deactivates your immune system. Where I think we talked about this early on, where there's this guy Wim Hof, and you see him on YouTube, and he's this lunatic. But then, if you start to actually look into the research, he's been brought into a laboratory, and it's been shown that he has a method, this breathing method that he uses to activate corticosteroid production. And when he gets cortisone in his body, it's able to suppress his immune system function. And so I would say that they're pretty similar.
Yeah, I think they're also going to be a key. The whole cortico, what is it called? The hypothalamic pituitary.
The gonadal axis?
No, it's the adrenal hypothalamic pituitary, because cortisol is released from the adrenal glands normally. I guess what I'm trying to say is that in a lot of diseases, we're hearing more and more these studies coming out like, hey, if you're stressed out, you're more likely to get cancer or get sick or all these things, right? And the mechanism for that, what do we do when we hear these correlations? We're like, well, it might just be like correlated or something. Or maybe you're stressed out just because you're sick or whatever, and not the other way around. Except they're finding that this axis, in particular the cortisol axis, is important because it's actually dampening your immune system. If you're stressed out all the time, your immune system is being suppressed. What does that mean? Well, it means that you probably have system level cortisol holding things back, so they're not able to signal properly. And you don't really want your immune system suppressed 24/7. You might want it suppressed when you're going through a terrible adverse COVID experience on the short term, because you want to survive. But in general, it's not a good idea to just, and this plays out even with topical corticosteroids too. You get really, really terrible injuries can result if you overuse those topical. You can buy these things over the counter, I think, right?
Yeah.
Yeah, so you can just buy corticosteroids over the counter. They work great for poison ivy, um, stuff like that, itches, rashes, whatever. But if you keep using them, you can get really terrible results that'll make you wish that you had your original problem back instead.
He has pulled up a really disgusting
So I'm
photograph
just leaving
of
that up
just
on the
an
screen.
open ulcer on some poor woman's arm. Does that just, are there other compounds that would mimic it? I think that it's basically just the adrenal system.
Yeah, yeah. It's like kind of the yeah. It's a very broad based uh, immune. This makes a lot of sense actually. You want to have something that's connected to your experiential existence that is capable of suppressing your immune system temporarily so that you can get away from the tiger that's trying to eat you. Or let's say that a giant storm comes and wrecks your house. You know, we've been evolving for millions of years, and this system obviously works in that it allows us to keep our head cold at bay for a few hours while we put the roof back on our house or something, or get away from the tiger that's trying to eat us. And then we can rest up and hopefully sleep it off and let our immune system get back to work. And when we're in that rest and relaxation phase of our body rhythms, the immune system can get to work, something like that.
I think that it also has to do with the fact that when you are out taking care of something that's kind of stressful, you need to be able to put all of your time and effort into taking care of the thing that is hard and stressful. And so having a molecule that's able to go through your body and basically just switch off all of the things that you don't necessarily need to be running at this moment is very, very helpful. And I think that it's also why you see, I don't know if you've ever experienced this, but you can have a really hard semester or you can have a really hard chunk of time and you're working really hard and you're just keeping everything going, but you're stressed the entire time, but you're not really sick. And then the semester ends, you hand in your project, everything is done, and then you get really sick for a few days afterwards. And I don't think that that's a latent head cold. What I think is happening is that your entire body is basically now being repaired.
It's a massive inflammatory
you've just held off at bay all of this inflammation just by virtue of being stressed out, and then when you finally relax, there's a few days of just feeling crummy before you can get really back up onto your feet after everything's been cleared out. So this is definitely has.
So if you spent spring break sick last week,
Yeah.
that's probably why.
I think that it's also a really interesting aspect of medicine, which is that if you have a disorder of some kind, or sorry, let me take a step back. Our bodies are full of these molecules, these hormones, these signaling pathways that are able to help us in normal times. So if you are relatively healthy and you need to sort of get down to lessen your inflammation, you can go for a run. If you don't overdo it, you will elevate your cortisol, then it'll come back down and you'll sort of reset the entire system. But if you're really sick, you lose the ability to do that. And as soon as you lose the ability to do that, then it makes sense for modern medicine to be like, well, we've actually isolated the compounds that can let you do that while you're convalescing in bed to get you back up onto your feet. And that's kind of the best and greatest version of medicine, which is that we have stuff that we know that it works and it works really well, and we can apply it for these short bursts and get people back up on their feet. That's kind of the great victory of modern medicine, I think.
Yeah, and we have to get some lumps on our head to figure it out. It seems like these miracle cures get overprescribed usually initially, and that's how it was with antibiotics, and that's how it was in the 50s with corticosteroids too.
Mm-hmm.
Until they developed NSAIDs, they were just prescribing corticosteroids for everything that everything from headaches to common cold, and it was just tearing people up, it seems like.
Wow, that's wild. So yeah, this is a great way to kind of monitor your own existence, where if you notice that this is starting to happen to you on a regular basis, you might be sick a lot. Like you might not be dealing with stress in a healthy way. You might need to have more intermediate periods where you find ways to decompress from everything that you're under in order to be able to let your body regenerate itself. Otherwise,
Exercise works well.
Exercise does work really well. Just walking.
Dancing, music, meditation.
Anything.
Whatever makes you happy.
Very briefly, this is a graph of all of the different ways in which these glucocorticoids are downregulating inflammatory processes and upregulating anti-inflammatory processes. Your T regulatory cells, if there is a seesaw of the way that your immune system works, where on one side it's inflammatory and the other side it's anti-inflammatory, your Tregs are the anti-inflammatory side of things. When you lift up your Tregs, it cools everything down and everything is much better, much more stable. Everybody else is basically responsible for producing all of the compounds that are going to kick your immune system into really high gear. And so glucocorticoids are global. They just inhibit pretty much everything except for Tregs and macrophages in low doses. What else we got here? Prophylaxis.
The corticosteroids, again, they're the only thing that absolutely work according to the meta-analyses we looked at.
Mm-hmm.
So it's probably worth all the time we just spent on them. But they are not necessarily the miracle cure in the long run.
And so now what we're going to do is we're going to go through some of the things that were indicated as being effective in the paper as prophylaxis, and we're going to talk a little bit about the mechanism of action. So
Talk about one of them.
which was the
these
names, Ritanovir or something?
Go back. I'll find it. We just talked about it.
It's this one. Ritanovir.
Ritanovir and Lopinavir. I wonder who comes up with these names. Ritanovir, yes. So that turned out to be a hopeful, but it's actually a prophylactic.
Somebody just asked if burnout could be considered a milder reaction to high stress. It seems like studies show that burnout was associated with increased systemic inflammation. I would say that burnout is basically the highest level of stress possible, to the degree that you have now saturated your receptors to the degree that your body's incapable of reacting to the stress hormones. Mm-hmm. Where normally the stress hormones are a signal where it's basically, I am stressed, I have to do something to fix this stress. And it's a motivating thing. It's energizing. It moves you towards the goal. If you get to the point of burnout, what's happening is that your body is dumping these chemicals into your bloodstream and your cells are just, I don't want to do anything with this. And so it's still having the bodily effects. It's attempting to have the bodily effects, but it's no longer able to have the anti-inflammatory effects because everything is saturated to this degree. And so I think that what's happening is that it ties to depression and this inability to do anything, right? When you're burnt out, you just sit and you're just wiped and you don't do anything. And so there's not this ability to get all of the fluids moving, to clear your body out, to get the lymph circulating again, to get the blood circulating again. And then once you start doing that, obviously, unless you have something clinically wrong with you, right? Like depression and terrible mental processes happen to people because they have genetic predisposition. They have things that can't be fixed with exercise. But if you do all of these other things and then the problem is gone, well, then you fix the problem for yourself. With favipiravir, this is a RNA-dependent RNA polymerase inhibitor. So what is it doing? It basically is coming into the cell, and it is, like the other drugs that we've talked about, attacking replication. An RNA-dependent RNA polymerase is one that is only active in the presence of RNA. Favipiravir is able to basically get into the active site of this RNA polymerase. It jams the works. It looks a lot like a DNA nucleotide. I don't have a picture of a nucleotide here, but imagine if you get rid of this fluorine and you get rid of this carboxyl group with this amine on it, then you
like the sugar backbone.
Yeah, it's got the phosphate sugar backbone, and that's the handle that the RNA polymerase is going to be using to sort of maneuver this thing. And
unsurprisingly, it works against a bunch of viruses. It seems like polio, Zika, equine encephalitis, seasonal pandemic influenza, rabies, Ebola, or I think that's supposed to say adenoviruses. Maybe not. And Rift Valley fever.
So again, broad spectrum, very effective, very safe, very cheap. A lot of this paper made the point over and over again that they're like, look, vaccines are great, but they're not always going to be accessible to everybody, especially because distribution is really tricky with these new vaccines that have to be refrigerated. The transportation is very difficult. Making sure that they get out to the corners of the earth is very hard. And we have this collection of drugs that work on other stuff. Let's look at it and let's use them and let's figure out how to make something useful out of it. And so favipiravir is one of these things that's just, it's worked a lot. It seems like it's promising for COVID as well.
91% of treated patients had improved pulmonary inflammation markers, compared to 62% in control group. Yeah. It says it's in phase 3 clinical trials here. And it says there's a Canadian phase 2 trial with this right now. I don't know how old this is.
So it's
Last
moving.
year.
All right, so this one is ivermectin, and I pulled, I basically went through and I pulled all of the ones that the study was suggesting were more or less effective. And at this point, we're in the repurposed drugs section because a huge controversy over ivermectin has been the fact that, oh man, this is not a drug that is an antiviral. It's an antiparasitic, and the way that it works inside of parasites is that it binds to a calcium channel preferentially. It floods the nematodes with calcium, causes paralysis. Eventually they die. When they die, they can be cleared from the system. And everybody was basically like, there's no mechanism by which this ion channel inhibitor should be functional for COVID. What are you talking about? And then there's this paper from Egypt.
Well, it seems like it has antiviral abilities against a bunch of other viruses too, historically. Chikungunya, I can't say this. Chikungunya?
Mhm.
Yellow fever, West Nile, Venezuelan equine encephalitis, dengue, and influenza.
So there's definitely, but I think that the complaint has been that there's not a clear mechanism of action. People don't really understand how it's working. And so I like this figure from the Egyptian paper because they're basically saying that, like, look, once the virus gets into the cell, it starts to produce proteins. One of the proteins that it produces is this nucleocapsid protein. And the nucleocapsid protein binds to a series of basically shuttle proteins. And the shuttle proteins brings it into the nucleus where it's able to continue its genomic assembly process. It binds to the host DNA and it deactivates a tremendous amount of host genes. It is stopping protein translation by interacting with the ribosomes. And ivermectin basically comes in and it seems like it interrupts the ability for the nucleocapsid proteins to interact with this shuttle protein, which means that they never get to the cytoplasm, they never get to the nucleus. If they never get to the nucleus, they're not able to actually induce this global gene expression change, which is what they're responsible for otherwise. They also think that what ivermectin is doing is that it is preventing the virus from being able to activate TNF-alpha and IL-6. And again, this is a little, this doesn't really have a hard mechanism here. This at least is showing that there's some kind of shuttling process. But the presence of the virus is capable of activating these inflammatory markers, transcription of them. And so if ivermectin is able to prevent the virion from getting into the nucleus, it could be that it's preventing the production of TNF-alpha and IL-6 at the nuclear level. It could be that it's preventing it from being able to be sent out of the cell at the cytoplasmic level. But the end result is the fact that it appears that in certain cases, taking ivermectin can decrease the cytokine storm, which is so lethal to people once you have severe disease. This is your death cascade.
But it only seems to work very, very early on, if at all. They cite in the paper a study of healthcare workers in India associating 73% reduction in infection rates for those pre-treated with this agent.
And that's another thing with clinical trials, right? If your substance is a prophylactic substance and you have to treat well before somebody encounters COVID. You have to start really, really early in order to get that treatment to them, and that can be very hard to do. Interferon. This is one of my, I think that this is the favorite figure that I pulled out from everything, which is basically you have, this is the apical surface of the cell, which is exposed to the interior of the lung. This is the basal level, which is exposed to the interior of the body. And interferons do exactly what they sound like. Interferons interfere. And so normally what happens is that you have this dendritic cell that's hanging out here. It's kind of partially in the basal membrane. It's partially up sentineling around the epithelial cells. And normally what you have is that interferon is produced by either... Interferon is present in the body, and when it is received by the cell, it is able to activate various interferon-stimulated genes, and it also activates the production of more interferon when it senses that the virus is there, and these interferon-stimulating genes are able to stop viral replication. And so if you don't have interferon, like for example, early in the pandemic, there was a BBC study that basically said that interferon mutations were responsible for severe COVID. If you don't have the ability to produce normal interferon, you're going to get either an inefficient interferon 1 response, or you're going to get an excessive inflammatory response, where insufficient interferon response is just not enough of it. You're not activating these interferon stimulating genes, and the virus, as it replicates and it mounts in number inside of your cells, is able to actually go into the nucleus and stop the production of these safety genes. And so then it runs out of control. The same thing happens if you don't have enough interferon and you're not able to bring down this inflammatory response. You get massive inflammation, and viral replication is running rampant because your body can't tamp down the inflammatory response, and it's busy dealing with everything being on fire rather than focusing just on this virion. And so giving people interferon seems to help. Do you have data on how much it helps?
No, I mean, I can pull it up. There's, well, we talked about interferons in one of our other lectures too, so we should go back and check that out if you haven't already.
We'll come back to the interferon question. It does seem like
there's no reporting. They're at phase 2, phase 3. Yeah.
They do seem to think that giving people interferon is going to be very helpful.
70 in a study from China, efficacy of interferon alpha 2b in 77 patients found that the treatment led to a significant decline in viral load in the upper respiratory tract, and it decreased IL-6 and CRP, which is a broad-based inflammatory marker.
Mm-hmm.
Yeah, not a lot of data from this paper on that one. Let's keep cruising, though. We got a bunch of these to get through.
Yeah, I'm not going to do all of them. So I have bromhexidine hydrochloride,
That's a cool one.
which is a cool one because it's basically just this chemical. What was the function of it? Oh, it's an expectorant.
Oh, yeah.
It's basically something that helps you cough out mucus. So I imagine...
it interferes with that. We've talked about this in one of the other classes, too, with attacking that TMPR SS2 enzyme, which is the native... it helps the spike protein dock and unfold the ACE2 receptor.
Yeah, it's a protease. Exactly.
And so we kind of asked some questions earlier about whether that could be a target of drugs, and here we are.
There we are indeed. And so basically anything that you can target in small enough amounts for a brief enough window might work as a drug treatment, because if you can stop this binding and you can stop this proteolytic cleavage, you stop the virus from being able to get into the cells. It's not harmful to you if it's not inside of your cells because it can't replicate. Colchicine.
It's also like a cheap, like, over-the-counter drug, it seems like. So that's really cool.
Yeah, colchicine is also one of those things that basically holds down the immune system. What it does is it interferes globally with the inflammasome. And so when colchicine is around, it prevents the production of all of these TNF alpha, IL 6, IL 1 beta, IL 18, super pro inflammatory cytokines. And it also prevents the recruitment of neutrophils, which are these basically bombers that show up and destroy tissue. And so if you can apply it at the right time, you can have a pretty serious positive effect.
Yeah. So I think that that's basically through. We didn't get really to the supplements. I want to make sure we have time to discuss
Okay.
these
Yeah, let's.
matters.
Did you have kind of like an overview of supplements quickly? I think I still have it open.
No, but I mean, let's upload these slides to the Moodle after the class. We can throw them in the description too for the lecture. Something, a link to them somehow, or at least a link to this supplements, because this is really fascinating. The bottom line though is that the CD or is this the NIH? The NIH isn't going to make any big claims about it because the studies are underpowered. Largely because there's probably not huge industrial funders that are going to pay for, multi-million-dollar studies on these supplements. But there's all kinds of really cool anecdotal evidence for each one of these, and at least the NIH seems to actually go through and say that most of these things don't really have, echinacea is safe. Does it work? I don't know, but it's at least not going to hurt you. So most of the things on this list fall under that category. And they range, a lot of them do have literature here. For instance, magnesium is going to help you out a lot with other processes in your body. And if those and that's been more well characterized than if its efficacy in COVID specifically. So a lot of these supplements might help you strengthen your health overall, which probably is going to help you out because we know that if you're healthy to begin with, you'll probably have a much greater chance of not encountering severe adversity with COVID. And so this is a really cool list. We just don't have time to go through it all right now, but quercetin's one of my favorites. Selenium's getting a lot of attention right now. Just all of these things, and you can find them in foods. So you can go through this list and be like, what kind of foods can I eat to maximize my intake of these different micronutrients and vitamins? And chances are you're probably eating a lot of them already. But yeah, I think it's pretty cool.
So this is more offered not as a point of discussion, but as a resource, because pharmaceuticals are best applied in dire straits. Prophylaxis is what allows you to maintain the state that you can call quote unquote healthy. And so if you look through this list and you see that there's stuff that you can add to your diet or things that you don't have, you can also, if you have student health, you can go get a blood test and see if you're short on any micronutrients. And being able to kind of monitor your own health like that allows you to be more proactive about it rather than just waiting for the moment that things start to fall apart and then you need help.
Like zinc, they've shown in lots of studies that it helps prevent viral infections, all sorts of viral infections. Have they shown it in COVID? Not yet, necessarily. And that seems to be the case with a lot of the stuff on here. They're like, we're sorting it out. But, if I had to roll the dice, I would say probably will help you out because it helps in so many other things. And there's a lot you can do with your diet to really strengthen yourself. I'm just a huge fan of keeping the body strong in the first place. And yeah, without really preaching about that, check it out. This is a really cool resource. I'm going to dig into it myself a little bit more later. With that, let's take a little break and then we'll field discussion and questions and curiosities and see what y'all are thinking about all this.
Let's do it. 5 minutes. We're back at 31.